Today, our presenters, Lisa Bramer and Kelly Stratton, are here to introduce the Multiomics Analysis Portal, or MAP, which is a one-stop omics data analysis web portal for EMSL users. It can perform quality control, exploratory data analysis, visualization, statistical analysis, and data integration. They'll go over the types of omics data that MAP accepts, what the different tools within MAP can do, and additions coming to MAP later this year. At the end of the presentations, we're going to have a Q&A period, so we invite you to post your questions in the chat. We'll pull those and we'll have some time at the end to go over them. So with that, I am going to toss it over to Lisa Bramer to get us started. Thanks for being here.
Thank you. Heather, sorry, I didn't get my camera turned on before. Oh, hold on, I got it. Perfect. All right, thank you everyone for attending today. So today, Kelly and I are going to discuss the Multiomics Analysis Portal. Just a quick introduction. So I'm a statistician, data scientist here at PNL, and a team lead for the data science and biostatistics team in computational biology. And this portal is the result of a couple of proposals that I wrote within the EMSL S&T portfolio. And for those of you who are familiar with the R package, PMART-R, I along with myself and Kelly are the creators of that package. Kelly will join us in just a bit here. She's a biostatistician here at PNL, and she also is an EMSL integrated research platform lead for data transformations. So today's objectives, I first want to go over the overall concept of what MAP is, what types of analyses and data types are supported, how you can upload data into MAP, go over some of the current applications that are there, and then talk about how to get involved. So the EMSL user program and EMSL in general stewards many different instruments, many of which you see here, all the way from the world-class 21 Tesla FTICR instruments to NMR and other types of mass spec-based omics. And as you can imagine, this creates a lot of data, and a lot of you have user proposals where you're generating these types of data, or you may be generating similar types of data at other institutions. One thing to note is that when we generate this data, first, hopefully you are engaging with a statistician at the beginning to do some experimental design, think about the response that you care about. You collect those samples get run on one or more instruments, and some sort of molecular identification happens. And perhaps in some cases also the intensity of those molecules or relative abundances are also measured and quantitated. And where we pick up is after you have this table in the bottom right of different data types with an identified biomolecule and some sort of either presence, absence, or quantitated intensity for that for each of those molecules. That's where we're coming in. So just as a heads up that we're not attempting to do any of the molecular identification itself. There are other tools within EMSL and outside of EMSL that can be used for that. Where we'd really like to come in is you generate a bunch of data from your experiment, and in order to really make the most out of that, you have to have a complete suite of expertise, meaning you have to understand the biology or the chemistry of what you're studying. To really dig into the data, you would have to know how to do statistical programming, and you also have to know what statistical methods are appropriate when, and all of that can be really overwhelming. So our goal is for you all to get your hands on your data as soon as possible and remove some of these barriers, particularly the coding and having to know exactly what statistical methods are appropriate when. And we were proposed to do that using this Multiomics Analysis Portal. So part of the concept here is EMSL in particular is generating very similar types of data. So you may run your data and get metabolomics and proteomics. And these data types are very similar from the spreadsheet type of view where you have biomolecules and samples and you get some sort of information about those. But the challenge here is that even though a lot of the data types are very similar, they may require very distinct types of analyses. So, for example, if you run NMR data, we know that that's an actual quantitative concentration of metabolites. If you're running proteomics data, those are in general for bottom up, that's a relative abundance, right? If you have RNA-seq data, those are counts. In proteomics data, you get lots of missing data versus metabolomics or lipidomics, you may have very little missing data. In addition to that, everyone has their own type of experimental design, whether you're comparing two groups or you have a time course, and the goal at the end of your analysis may differ. So keeping that all in mind, the big idea here is that rather than trying to build one giant application that handles every type of data, every experimental design, every type of analysis, we built the Multiomics Analysis Portal, or MAP, and it's designed to be more like an app store like you would have on your phone, right? So the benefit of this is that we can have distinct types of tools that are useful for certain types of data or certain types of experimental design or certain analysis goals. And we can easily add new tools as new instruments or technologies are being brought online. And we have this backend framework that allows you to pass data from one application to another. So what I'm showing right here right now are some of the applications that are either currently in MAP or set to be released in MAP very soon. We're also in the long run looking for, as you hear about the tools that already exist in MAP, if there are other types of data analyses or analysis goals that are of interest to you, we're also interested in hearing about what is the next thing that could be useful to you. So just very briefly, I'm going to go through the underlying framework of MAP. So MAP is a Shiny app, which is built in R, and it is built and Dockerized in a modular way so that we can deploy on the cloud, and then within that Shiny application are a bunch of other Shiny applications that can be called. And so in this orange box here, that represents MAP. You have a bunch of other Shiny apps like Frida, PMART, IPMART, and Mode that are the front end sort of applications in your app store that you can deal with and interact with. And in the back end are R packages that are actually doing the heavy lifting. The benefit of this structure is that when you go into MAP and you launch an application, you get your own instance on the cloud, so you should not be inhibited by other users or the load of how many users are actually accessing the application. So with that, I'm going to pass this off to Kelly Stratton, who's going to walk you through some of these applications and Mode, how to upload your data, or MAP, how to upload your data, and what these applications look like.
Thanks, Lisa. So I believe that the link for accessing MAP has been dropped into the chat or will soon be. And when you go to that link, you will be actually prompted to log into Nexus, which is EMSL's user interface. And the reason for that is this deployment in the cloud. So you can request a Nexus account if you don't already have one. This is, a free resource, but it is the way to access MAP. And the current state of MAP is that you need to upload data in the form of CSV files and create a project. And you're able to access the data that you've already uploaded in later sessions, although this isn't persistent. So at some point it will be cleaned up. So if you want that persistent data access, what you can do after you've uploaded and even analyzed data is you can then download a MAP project file, as we call them, either to store yourself or to share with colleagues so that they can upload those and run through an analysis or access the same analyzed data themselves. In the future, our plan is to actually connect MAP to Nexus so that that step of needing to upload data is sort of taken out of the loop and it's more seamless. So look forward to that in the future. I want to talk a little bit about the different analysis types that are available and the status of those tools. So the first one here is called PMART, and this is the Panomics marketplace where you can do quality control, statistical analysis, and visualizations associated with those processing steps for a single omics data set. We also have IPMART. The I here is for integration. So this is essentially PMART for more than one omics data set with some built-in integration tools so that you can bring together, say, your metabolomics and your proteomics data. We also have MODE, which is our multi-omics data exploration tool that enables you to interact with data visualizations and sort and filter through statistical results. In addition, we have FRIDA, which is for exploratory analysis of FTMS metabolomics data. And Lisa will be showing a demo of that in a little bit here. And then the last on this list is SLOPE, which is for guided machine learning. So of these 5 apps, we have 3, the top 3 are available now. PMART has gone through a round of beta testing. So we're looking for additional users there. And IPMART and MODE, we are in the process of recruiting beta users. So be thinking about whether that's something you might be interested in because we would love your help. FRIDA will be available soon. Updates are currently underway on that tool, and SLOPE is being actively developed right now. So as far as the different types of data that MAP can work with and analyze, I've touched on some of these. So there's FTMS metabolomics data, GC and LCMS metabolomics, LCMS lipidomics, label-free or labeled proteomics data, NMR, as well as RNA-seq transcriptomics. And as we've sort of alluded to already, the thing that we have done in the development of MAP is make it so that there's a really consistent format for these data files that currently need to be uploaded to MAP, regardless of the type of data that you're working with. And I'll go into the format of the data in a couple of slides. But first, I want to sort of make the bridge between the different tools and the types of data that they can handle. So PMART, IPMART, and SLOPE are all designed to work with all of the data types except for that FTMS data. And MODE currently will handle all but the RNA-seq and the FTMS. FRIDA is solely intended for the FTMS data because of the nature of the data and the types of analyses that are appropriate for it at this point in time. So what I'd like to do now is sort of give you a brief tour of the MAP tool as a whole. So we have a homepage where you can access documentation that will tell you about the data that you need to upload and what that should look like, and about the different features that are available in MAP. There are also links to some publications and a GitHub repository for the underlying PMART-R package. The data upload tab is where you can select the type of data that you will be working with and actually ingest data files. You can then manage and view the different data that you have uploaded so that you can come back and work with it later, see which tools are appropriate for it. And then if you want to explore the tools in a little bit more detail, you can use these filter drop-downs to see which tools are appropriate for single data sets, multiple data sets, the types of data that you've uploaded already into MAP, and even analysis goals. You can also use this design a workflow section to pick a set of data that you've uploaded and see what different types of workflows are available within MAP. So you could start with your uploaded data, process it in PMART or IPMART, and then send that into mode for some interactive data visualization. So here, oops, next slide. There we go. I'd like to talk a little bit about the data upload process. So the first step to doing any type of analysis in MAP is to create a project on that data upload page. And this involves selecting the type of data that you are going to upload. So whether it's label-free proteomics or NMR, assigning a project name of your choosing, and then actually uploading 2 or 3 CSV files. So here on the right is a diagram showing the 3 different types of information that are required, with this bottom one actually being optional. So at the top, we have our expression data. So this is a matrix of values that you get from the identification and annotation that Lisa was talking about. And the structure of this file is that you have one column for your biomolecule identifier. So here it's showing peptides. And then the remaining columns are associated with each of the samples that were run. So here you can see that we have numeric values as well as some NAs, and that those are representing missing values in this data set. The second required file we refer to as the sample information file. And so this maps the sample names that were in your expression data across the top to different values associated with the experiment. So here we have our sample names. We have a group, which in this case, control and experiment. It's likely going to be much more tailored and specific to your study, but this is just indicating, we are interested in comparing between these groups. If you happen to have paired samples, so perhaps you are taking a measurement before and after some sort of intervention, you can indicate that in this file, and any other information that is pertinent either to what you might be doing in a PMART analysis or just that you want to be tracking along with your samples throughout the data analysis. This third file, the biomolecule information, is optional. Although depending on the type of data that you're working with, you might always, utilize this file. So here in our example with proteomics data, we have our peptide column, which matches that in the expression data file at the top. But we also have this mapping from peptide to protein. So in PMART, we can actually roll up from the peptide level to the protein level so that we can do statistical analyses on the proteins rather than the peptides. So in order to do that, we need to have this information in our biomolecule file. You could also include things like whether a protein is a contaminant. For lipids, you could include lipid class information. You might want to map to different ontologies or databases to look at different types of identifiers for the same molecule. So here, I would like to show you actually uploading data into MAP. So the first thing that we're going to do is choose the data type. And for this example, I'll use label-free peptide data. and give it a project name that is meaningful to me. And then after I click that use project name button, I can select my files for upload. And you can either do that one at a time, or you can select multiple at once. And then the next thing that needs to be done is to associate those uploaded files with the type of file that they are in MAP. So here, this PEP_E data is my expression data file. And I'm also associating these files with this project that I just created. So I do the same for the biomolecule information and the sample information files. And then I can save that project and submit it to the server. And now if I go to manage files or to the map store, I will be able to locate my project. So here in the map store, if I go and click on PMART, I can select that uploaded data, peptide example, and then launch the PMART app. And it takes a moment for it to load, but what it's doing in the background is actually pulling the information that it already knows from my submission into MAP, right? That it's proteomics data, it's unlabeled, I have peptides. And it's starting to populate the PMART app with those values. So it knows, it's guessing actually that that first column peptide is my identifier. And then I can tell it that I have raw intensity data and I want to transform it to a log scale. I can also go into this biomolecule information section and choose the column from that file that corresponds to my protein identifier. And once I'm happy with those selections, I can create the omics object and then go to my group samples tab, which is where I'm dealing with that sample information file. And so here I can choose the column of that file that I want to be my basis for comparisons later on, which is phenotype in this case. And then I can proceed through the PMART app. And here, what I'd like to show you is just one of the plots that we often make, which is just box plots, one box per sample. And I can choose to order and color those boxes by some property of the samples. So here I've selected phenotype. And when the plot generates, I'll have these 3 phenotype groups. I can also use this Save Last Plot button that's at the top of the screen. And we can see it's kind of incremented my plot count for view all saved plots to 1. And what this does is it saves this plot in its current form for me so that I can download it later. And I'll show you more of that in a little bit here. So I'm not going to walk through all of the steps of a PMART workflow. I'm just going to show you a few key portions of it. But if this is something that you're interested in, stay tuned because we have some options at the end of our talk today for getting involved in using the specific tools available in MAP. So the rest of the PMART workflow consists of additional summary visualizations, like the ones that are shown here, a PCA plot, some summaries that show missing values by sample or by biomolecule. We have the ability to filter our data, either to remove biomolecules or samples, to normalize our data. For proteomics, we can roll up to the protein level, as I mentioned, and then also perform some statistical comparisons. So that's actually what I would like to show you here. Once we get through those intermediate processing steps to the statistics tab, we could choose to make a PCA plot with our normalized log2 protein data, which is shown here. We could also select the other option, which is IMD ANOVA, and this will actually let us do comparisons either between the quantitative values with the ANOVA or based on the presence absence of a protein with the IMD or G-test option. I've selected which comparisons I'm interested in, and there are some settings for the statistical tests that you can choose to include or adjust. And then you can perform those tests and see different visualizations of the results. So we have volcano plots. Again, we could save the plot so that we can download it later. We can actually adjust the way the plot looks by changing the colors, the font sizes, the style of the axes. So if you're going for a specific look for your plots, there's the ability to make changes to those before you save it. Go to the next slide. It just wants me to redo there we go. So when you have gone through and performed statistical comparisons, the last sort of step in the PMART tool is to save output from your session. And you saw me save some of the plots that were made along the way. And it's at this last stage that you can download those plots that you have saved. You can also, so this top portion of this screen is showing just sort of the folder of all the things I've collected along the way through my PMART analysis. So I have these plots 1 through 5 that I had saved. I also get back my biomolecule information file, my sample information file, and then I get 2 versions in this case of the processed expression data. One is at the peptide level, and then the other is at the protein level after roll-up. And that protein expression file, just a snippet of it is shown here in the bottom right. We also get a CSV file that contains the statistical results from what we did in the PMART tool. This table ends up looking rather similar to what's shown on the screen, where we've got a row for each of our proteins, and then we'll have columns with different results for the comparisons that were done. And that includes p-values, fold changes, means, and the number of observations for the various groups and comparisons. And on that last section of PMART, you can also generate a report that has recorded all of the processing steps that you went through, as well as includes lines of code from that PMART-R package that were used, so that you could go back, or you could have a colleague go back and actually recreate your analysis, either in the PMART tool or from within R, if that's where they prefer to work. So this will just sort of scroll through and show you the structure of one of these reports. I have the option of naming it something meaningful to me. It gives you the summary of the data that you uploaded originally, and it tells you what type of data it was. In this case, it was unlabeled peptide data. We transformed it. All of that information is included here. The filter section tells us which filters were applied and what the result of those filters was. So how many biomolecules were removed. We also capture information about the normalization approach that was applied. For our protein roll-up, it details the method that was used for that, as well as all of those options that we selected on the statistics section of the tool. It also includes some of the plots that were generated along the way from within PMART. But again, you have control over which things are saved so that you get a specific file for each plot at the end that you can download for a presentation, for instance. What I'd like to show now is what you could do with the output from that PMART workflow. So you can, in addition to downloading all of the things we just talked about, you can send the results from your PMART analysis back into MAP and actually open them open them up in mode for visualization. So here we can see we have this saved proteomics example. So when we click on mode in the map store, we can find that from our dropdown and then launch mode. And it goes through a similar process that we saw for PMART, where it's pulling in all of sort of the attributes that are tracked on the back end so that we have fewer inputs we need to provide in the mode tool. So what mode provides is a trellis visualization. And this, I'll show you in detail what this can look like. The way that it gets built using this tool, though, is it'll show you one plot. So here for our protein data, this is a plot for a single protein, and we can choose what type of graph we want to see. So whether it's abundance box plots, whether it is the proportion of missing values for that protein, or even the fold changes that we saw for different comparisons. And then we can actually generate a display where we have one of these plots for every single protein in our data set. And then we are able to, similar to PMART, adjust how these graphs look, what other information we might want along with them, which I'll describe that in a moment here. And then create this trelliscope display, as it's called, and then download it so that we can interact with it later. We can share it with a colleague and learn more about our data through visualization. So I'm going to steal the screen now. And you should be seeing a big box plot. So someone please let me know if that's not true. This is the display, thank you, that was made in mode that I have just downloaded to my computer. And I will walk you through briefly what this looks like. So right now we're seeing a single protein. We have the log to abundance on the y-axis, and each of our three phenotypes here denotes one of the boxes. So each of the points on top of these box plots is a single sample, and we're seeing the log to abundance for that sample broken down by phenotype. So we have 2,700 proteins in this data set, and what I did was filter down to the most significant of the proteins for just for the purpose of a smaller visualization here. What we can do, though, is change the number of plots that we see at once. So now we're seeing the first 6 out of those roughly 2,700, and we can flip through them. But where the real power of these displays comes in is in our ability to sort and filter these plots. So right now we are merely sorting alphabetically by protein name. But what I would like to do is change this so that we are sorting, let's sort by the phenotype 3 median abundance. So now we are seeing a different ordering of our plots, and we can flip through them, and we might start to notice different trends. If we, on top of this, add a filter, so we were sorting by the median phenotype 3 abundance. So why don't we hone in on phenotype 3 versus phenotype 1 fold change that is greater than about 1.5. So now we're down to just 4 of these plots. And we can start to see, okay, well, it makes sense if we're filtering on this fold change, right? That comparing phenotype 4 to phenotype 1, we're seeing a bigger difference in those boxes. We could also broaden this to get more plots and really start to play around with finding trends that look interesting from a biological perspective. So at this point, I think what I'll do is hand it over to Lisa.
Thanks, Kelly. Let me steal the screen sharing from you. So I want to just briefly demo the tool that Kelly mentioned called Frida, which is specifically for exploratory data analysis of Fourier transform mass spectrometry, so FTICR or other models. And I've already uploaded data just to save us a little bit of time, but what you can see is that this tool just requires 2 of the 3 files that Kelly mentioned. So the expression data here at the top are the different peaks or M over Z's where the peaks were observed. Every other column is a sample. And then for every peak, I have potentially have a formula assigned where a software like CoreMS can go in and actually make these molecular formula assignments. In this case, I don't explicitly have to have my sample information with me, although I could if I wanted to, but here I'm just going to tell it that the peak is the thing that gives each molecule an identifier, and then I can tell it you have to have either a formula assigned or for some combination of elements, the counts for each element. Here, I'm just gonna select formula and tell it that this MF column is actually my molecular formula column. You can see if that I were to click on elemental columns and then it has this specify button right now, it will guess at which column represents carbon, nitrogen, and so on. And right now, these 6 elements are currently supported. We're in the process of updating this application, so any combination of elements that you're interested in can identify through some identification software can be included. And I'm going to go ahead and process my data. And once it's done, if I review the results, it tells me that I've uploaded data where I have 27,000 peaks, 36 samples, and about half of those have formulas assigned to them. I can then move on to the groups tab, and I can define some groups that I'm interested in comparing. And again, in the newest version of this application, we will also have the ability that you can upload one of these files that has information about your samples. But this particular data set is about soil samples that were transplanted from the bottom of a mountain to the top of the mountain to simulate climate change. So I can name my group, and here I'm going to call this upper native. And if I go down here to look at my list of samples, you can see I have all 36 samples listed here. That would be pretty tough to just go through and like hand check each one that I want included. So I can, if you name your samples in an intelligible way, I can go in and search. And I happen to know that everything that I'm interested in the sample name has a UN for upper native. When I do that and then click on my samples to include, you can see it's just searching and finding those sample names. I can select all of those samples and add a group of samples I'm interested in that pops up over on the right. So I'm going to actually also populate one more group where it's the soil samples that go from the upper part of the mountain to the lower part of the mountain. And I'm going to search sample names and also add this group. Okay, so I can continue to do that with all of the potential groups I'm interested in, and then I can continue on to pre-processing. And if you've dealt with high resolution metabolomics before, often things like elemental ratios are computed Kendrick mass and defects. There are other quantities that are sometimes computed too that we have programmed in here, and you can select whichever of those that you're interested in. You can click or just hover above a question mark. It'll tell you what it's calculating, or we also have a glossary over here that will help inform you. And if I go back to this and click process data, it's going to compute all of those values for me. And at the top, it provides me with a table of all the things that I've calculated and some summary statistics, as well as if you wanted to just see, you know, what does my distribution of oxygen to carbon ratio look like across all samples as a sanity check for your data, just to make sure everything looks okay. And once we're happy with that, we can move on to the quality control tab. And it'll take just a second here for this to pop up, but what's first being generated are just some basic plots about your samples. So for example, here, right now it's plotting how many peaks were observed in each sample. I can also look at the data quantitatively in terms of box plots. And in this case, if I were only interested in the two groups that I previously created, I can subset down by clicking them here. And see, these are the samples that I was interested in. And again, I can do just like Kelly showed with PMART, I can do save the last plot. And now it's populating a list of plots that I can download later. We can move on to the filter tab. So there are lots of different filters that you might apply, and it shows you how many peaks you have before and after filters are applied. It will provide you with some quantitative information, graphical information about what's happening. So for example, you might have an instrument where you really only believe or confident in identifications between certain masses. So we could apply that filter. As soon as I check that button, you can see that it shows me my unfiltered data and what the implications of filtering out anything that doesn't have a mass between 200 and 900, that I'm down to 23,000 peaks. Or for example, I could filter out peaks that have to be observed in at least 2 samples. And now I'm down to 12,000, right? So there are also ways of adding custom filters. So if, for example, you're only interested in looking at peaks that have certain oxygen to carbon ratios or certain aromaticity values, you can filter that down so you can explore those. You can always go back and reset your filters to take them off and filter your data in different ways. Just really depends on what you're interested in. And once I click the filter page, the power of Frida is really about trying to explore graphically what's going on. So one example is a Van Krevelen plot is a very common plot that's used. I might want to make that plot for a single sample or multiple samples, or maybe I want to compare 2 groups. And I can select the groups either upper native to the upper to lower. I can rename this group up here for plotting purposes. And I can actually do a statistical comparison to ask for which peaks is the peak present statistically significant more frequently in one group than another. And if I click generate plot, it's going to go through and do all of those statistical tests for me. And here the purple peaks are things that are observed in both of my groups. If I use this interactive graph and click off of it, you can see it. Now I'm left with things that are unique to group 1 or my upper native, or unique to group 2. And I can start to get a feel for if there are any trends in my data. I can also make single sample plots. So I'm just going to do that really quick. So let's say that this particular sample is what I'm interested in. I can make a Van Krevelen plot for that. Right now it's using some computed boundaries from a publication to kind of classify the different metabolites. I can go ahead and save that plot for later. I could also make a Kendrick plot using that same single sample. And here things are being colored by the number of carbons, but I could come and color my points by any of those properties that I've calculated. Save that plot. The other really neat part of this tool is that we have the ability to link plots. So amongst the plots that you've saved, if I wanted to click on both of these and compare these plots, so I've created plots for the same sample. And if I find some points that I'm really, kind of interested in, let's say these points over here, I can lasso those points. And what you can see is on the left, it populates a selected button. If I double click, it will just highlight those points, and I can see where the points on the right fall in the plot on the left, in case you want to be able to connect information. So without going through anything more, so that we have time for questions and everything, I'll just briefly say too, we have the ability to take those formulas that have been assigned and map them to to databases like KEGG and Metapsych. And similar to what Kelly has shown before with other tools, we have this download page where you can select the data, the process data, you can get a report of what was done, you can download the plots that you saved along the way. So back and someone could either recreate your analysis, or you have something that is capable of, like a figure that's being capable of going into a publication, where you can select the format and the height and the width. So with that, I will pass it back to Kelly. I guess actually first I will quick not pass it back to Kelly. So where are we and where are we going? So current work is that this Frida tool is going to be added into MAP along with PMART and IPMART, which are already there. We are currently working on a tool that's called SLOPE, which stands for the statistical learning of predictive evaluation. So basically, this is a tool where you can implement machine learning methods on your omics data. So perhaps either classifying samples and seeing what are the important biomolecules that allow you to classify those, or investigating which biomolecules are related to one another. And I do want to mention that what I showed you today was me uploading data into Frida. There will be a direct connection between MAP and CoreMS, and CoreMS is a tool that could do identification for your metabolomics data that would allow it to be directly sent to the Frida tool. So now with that, I will pass it back to Kelly.
Thanks, Lisa. So yeah, we just wanted to conclude with an invitation for any of you to contact us if you're interested in using PMART or being a beta tester for either Mode or IPMART. You can reach out to us directly. Also, our vision here is that we would like to hold trainings to focus on a single tool at a time, so that you can learn in the first half sort of how to go through a typical workflow with example data. And then the second half of those trainings would be an opportunity for you to bring your own data and work through it, ask questions along the way, and sort of get that real-time feedback so that you can learn by using your own data and doing your own analysis. There's a link here for a Google form where you could indicate your level of interest for these activities if you're interested in any of those things I just mentioned. And then also, if you have thoughts about other types of analysis methods that would be really helpful, or other types of data that you work with that you think could fit within the MAP toolbox, we would love to hear about that. You can reach out to me or Lisa if you have thoughts there. So thank you very much. And yeah, we're happy to answer questions.
Okay, Great. We have a fair number of them, so we're going to see how far we get in the next 10 minutes. And just please rest assured that if we get to one and we haven't answered your question, that Kelly or Lisa will reach out to you. Oh, yes, we have a question. Can you share the Google form for the training in the chat? Megan, I'm going to put you in charge of dropping that so that it can be shared. Okay, one question from Ravita. What about LCMS, MS data? What about LCMS metabolomics data? I believe that this is in reference to, at the beginning, we were talking about which apps support what data type. And this particular person is saying that they have GCMS profiling data and DNA sequencing data. Is there an ability to analyze these data together? So kind of two parts.
So the LC-MS data should be fine to put through our applications. I will say that particular data in the context of metabolomics is a newer, the LC-MS, sorry, is a newer technology. So we are currently working to make sure that if there are any nuances to that data that it can be handled. But as far as I know, we've had people actually go through and use that data in these apps. And if you have the other two types of data, you said were DNA and
GCMS.
Yeah, so that should be applicable to a tool like IPMart if you wanted to try to integrate those. And so the tutorials that Kelly is talking about would be a great way to get familiar with that. Alternatively, if you visit any of the apps, we have YouTube videos that go through each of the pages and how to use those too. If rather learn on your own time than having to commit to a certain time window.
Great. To follow up on that question, Panchak is asking, is there support for targeted proteomics data sets?
So by targeted proteomics, I'm assuming you're meaning things like MRM, SRM. Those should all be fine to go through this. The thing that we don't currently support are any top-down proteomics, but anything that is bottom-up, either targeted or untargeted, is supported.
Okay. Will this incorporate the LEAP-R package for biological pathways activity analysis developed by PNNL and EMSL?
I think that is Jason McDermott and Sarah Goslin's package, Kelly. It doesn't currently incorporate that. There is a possibility that could happen. So I would suggest if you go to that Google form that Megan pasted in the chat, that's one place where you can indicate additions that you would like to see. So if we have enough people that want that, then we can talk to EMSL about getting that funded so it can be incorporated.
Okay. David Hoyt has a question. Have you tried to pair LCMS and NMR metabolomics data?
You want to take it, Kelly? I feel like I've answered everything.
LCMS and NMR metabolomics data.
I'm assuming it means LCMS metabolomics.
Right. I don't know that I have explicitly done that before myself. I'm actually not sure that I have seen a project come across my my desk that used both of those methods. I don't know if you've seen that, Lisa.
I haven't, but I feel like it would be a very interesting thing to try to put through IPMart.
Definitely.
Have example data sets, so it'd be really cool to look at those.
Okay, great. Question from Leah Johnson. Is this only available to use if you have EMSL data, or can you use it for non-EMSL affiliated data?
So this can be used for non-EMSL affiliated data, especially with its current state being solely as a upload of your own data. It's open for the public. The only requirement is that you have that Nexus login to access MAP.
Okay. And we did actually have a question about that, about how to set up an account. Do they need to email someone to get a username and password, or can they just create their account on the Nexus website?
I believe you can just create it from the Nexus website if you go there.
Okay.
Should be a
That's my
button.
understanding as well.
Okay.
Really, the only reason we're requiring a login is because it is on the cloud. If you open cloud things to the public, then bots and web scrapers can come in and use up a bunch of resources. So
Got it. Okay. Question from Rebecca. Can you upload data that is already log2 normalized and or with samples relative to each other?
So yes, you can upload data that's already been log2 transformed and or normalized in addition to that transformation. There's a way you can indicate that when you do that data upload. As far as samples relative to other samples, I might need a little more information to know if you're talking about, for instance, like labeled proteomics, where you've already done a normalization to the reference pool versus something else.
Okay. So Rebecca, I'm going to suggest that you connect with Kelly via email so that you can get some more information there. Another question. Can I use this for the microbial community analysis for species identification and abundances?
yes, you should be able to. The caveat I will say is that some of the typical analyses that you might do, like alpha diversity, beta diversity, are not currently included in any of these tools. But again, like if that's something that would be super useful to be included, we would love to hear that, something that we can put in. But to do some basic statistics of that data or to visualize the data, totally fair game. It just, I know that in that community, there are some very specific analysis types that get done with regard to, say, measures like diversity that aren't incorporated.
Very good. Let's see. How do you calculate the p-value for metabolomics data in the MAP portal?
So in that PMART tool on the statistics page, when you select the comparisons that you're interested in, so for the example that I showed, we had 3 phenotypes, and you run the statistical tests, that output file that gets generated, which you can download, will contain the p-values for all of the comparisons that you specified, either adjusted or not, depending on what you selected in the tool.
Okay, very good. All right, I'm going to make this the last question. And this is kind of a confirmation that they wanted to check on. Users can decide by themselves which comparisons should be made and can select the reference, generally the control sample for their analysis, right? Or do users have to change the metadata every time?
You don't need to change the metadata. In the tool, you can pick the comparisons and sort of the order of that comparison, whether it's phenotype 1 versus 2 or 2 versus 1, depending on what makes more sense.
Okay, great. All right, actually, I'm going to take one more question here because we've got 1 more minute. So we should get all of our time out. Okay, so will this be integrated with DOE's K-base predictive biology platform in any way?
As of right now, no. We have been talking with K-base folks about that possibility and what would need to happen. But I would say within the next year, the answer to that question is no, but it's possible in the long run.
Well, with that, we are going to wrap up today's webinar. Again, if you didn't get your question answered, I will share these questions with Kelly and Lisa, and you'll be hearing from them. Thank you for attending. Check out the MAP resource page on the EMSL website for some more information. And don't forget to join us back here on October 25th for the EMSL Learn webinar on the upcoming Monet proposal call. But thanks and have a great day.